Welcome to the final video for the Nutrient Cycle series. Here we will offer a comprehensive solution to Darwin's paradox. So far, I have introduced you to the microbes that can convert ammonia into nitrite, nitrite into nitrate, and nitrite and ammonia into dinitrogen gas. None of these microbes, however, hold the answer to our paradox regarding how new nitrogen is introduced into the system to prevent the system from running out of nitrogen as the result of the activities of denitrifiers. To be able to prosper, the coral reef ecosystem requires a continuous input of new nitrogen to balance the loss. Our atmosphere is 78% dinitrogen gas and therefore offers a plentiful source of nitrogen for the oceans through dissolution at the air-water interface. However, in this form, it is only biologically available to a specialized group of microbes known as nitrogen fixers or diazotrophs. Examples of diazotrophs can be found in both archaea and bacteria domains. All have evolved the ability to capture this inorganic form of nitrogen, dinitrogen, and convert it into ammonia, which can then be used either directly by other organisms or converted into different forms by other groups of microbes. There are examples of diazotrophs in every lifestyle type available to an organism, even including photoheterotrophs, which are organisms that can use light as their energy source, but they are unable to use carbon dioxide as their sole carbon source, instead using organic sources such as carbohydrates, fatty acids, and alcohols. And they may also be either aerobic or anaerobic. Because of their broad diversity of ways of living, they can be found in many different niches, both free-living and endosymbiotically. That is, living within the tissues of microorganisms where they provide a source of organic nitrogen directly to their host. Diazotrophs are exemplified by the cyanobacteria. Species of cyanobacteria are either unicellular, which are invisible to the naked eye, or filamentous, such as members of the genus Trichodesmium, that can occur in such high densities that they can be seen by orbiting satellites. Trichodesmium is colloquially known as sea sawdust because of the thick slicks it forms on the surface of the water. Although cyanobacteria are predominantly phototrophic, some species are able to grow heterotrophically under dark conditions. Their ability to fix carbon or consume organic carbon under specific conditions means that they play an important role in both the nitrogen and carbon cycles. Nitrogen fixation requires a large amount of energy, which they gain in a variety of ways depending on their mode of living, and involves the enzymatic reduction of dinitrogen gas to ammonia. This is carried out by a family of enzymes called nitrogenase. These enzymes are irreversibly inhibited by oxygen. To get around this problem, diazotrophs employ a range of strategies, including spatial, temporal, and metabolic modifications. Because cyanobacteria are photoautotrophs, they face the problem of producing oxygen, which will directly inhibit the nitrogenase enzyme and therefore their ability to fix nitrogen. Some filamentous cyanobacteria have got around this problem by spatial modifications in the form of specialized cells called heterocysts. These cells contain the nitrogenase and their cell walls are impermeable to oxygen, allowing nitrogen fixation to occur at the same time as photosynthesis. Filamentous cyanobacteria that lack these specialized cells, such as species of the genus Trichodesmium, use a metabolic adaptation, whereby oxygen produced in the first stage of photosynthesis is rapidly reduced to water, preventing the inhibition of nitrogenase. Other nitrogen fixers, such as those that live symbiotically within the tissues of reef-building corals, live in close proximity with endosymbiotic microalgae. This could cause a problem because during the day, the endosymbiotic microalgae are photosynthesizing, using CO2 to build sugars and producing oxygen as a waste product. As we've said, oxygen prevents nitrogenase from working. But at night, the macroalgae switch to cellular respiration, using up oxygen and sugars to perform their metabolic functions. In these low oxygen conditions, nitrogenase can function and fix nitrogen gas to ammonia. We call this temporal separation because nitrogenase activity and oxygen production occur at different times, allowing both photosynthesis and nitrogen fixation to occur in the same location. Now we have learnt about all these different microbes, we are able to go back and solve Darwin's paradox. The reason coral reefs are able to thrive in the oligotrophic waters of the tropics, where both nutrients and consequently also phytoplankton are limited, is due to the presence of a wide range of microbes in the water, on biotic and abiotic surfaces, and even living endosymbiotically within the tissues of coral reef creatures themselves. We can understand just how important they are for system function if we break it down into an atmosphere and the water column sitting above the oxic and anoxic sediments. If we only had plants and animals in the ecosystem, it would gradually wither away and die, as the cycling of nutrients is never 100% efficient. Nutrients expelled in animal feces are not always available for reuse, and as both animals and plants die and the carbon and nitrogen in their tissue remains locked up, over time this results in a loss of the accessible nitrogen in the system. The input of nitrogen to the system from the atmosphere would help to compensate for the loss and help to support the growth. However, the amount needed would be very large and require a very large number of nitrogen fixers to convert it into forms available to other organisms, which would also require a large amount of energy to carry out this process. Therefore, another source of nitrogen is required. This can be found locked away in the remains of dead plants and animals and in their fecal matter. Again, it is the microbes that are responsible for unlocking this precious resource through the decomposition of the dead bodies and fecal matter into bioavailable inorganic nutrients. They also purge any excess nitrogen back into the atmosphere by converting it into ammonia and other forms of inorganic nitrogen, and then into dinitrogen gas. Clearly, the balance between how much nitrogen gas is taken in from the atmosphere versus how much is lost depends on how much is required for growth, and also on the prevalence of plankton in the surrounding water column. Later, you will hear about the role of planktivores that hang about on the reef crest to efficiently consume any available plankton as it washes past. This brings us to the end of our final nutrient video. Here we have focused on the nitrogen and carbon cycles and have seen that microbes and the alternative modes of living that they use, other than those that one typically associates with plants and animals, are critical to the survival of coral reefs in the oligotrophic waters of the tropics. Thank you for listening.